Hello everyone, we are going to see about lymphoid organs. What are lymphoid organs? Lymphoid organs are usually concerned with immune reactions by means of the production, maturation and proliferation of the lymphocytes. They are called as the lymphoid organs. So, the lymphoid organs are very important organs in the immune system. They are concerned with immune reactions by means of the production, proliferation and maturation of the lymphocytes. There are two types of the lymphoid organs. Primary lymphoid organs, otherwise called as the central lymphoid organ. Secondary lymphoid organs, otherwise it is called as the secondary peripheral lymphoid organs. First of all, we are going to see about what are primary lymphoid organs, otherwise the central lymphoid organs. Primary lymphoid organs are the site of formation of the lymphocytes. The T lymphocytes and B lymphocytes mature into immunocompetent cells in the primary lymphoid organs in the absence of the antigens. Because the antigens cannot enter into the primary lymphoid organs because of its capsular structure, the antigens cannot enter. So, in the absence of the antigens, the T and B cells, they are matured into immunocompetent cells in the central or primary lymphoid organs. After the maturation, the lymphocytes migrate to the secondary lymphoid organs through blood and lymph. The primary lymphoid organs are thymus and bone marrow. These two are considered as the primary lymphoid organs. The T cells mature in the thymus and the B cells mature in the bone marrow. What are secondary lymphoid organs? It is called as the peripheral lymphoid organs also. These organs are those where antigen is concentrated and allowed to interact with the immune cells for proper immune response. These include spleen, lymph node and MALT. MALT is called as mucosa associated lymphoid tissue. This includes NALT, that means nasal associated lymphoid tissue, GALT, that is gut associated lymphoid tissue. The other one is BALT, that is bronchus associated lymphoid tissue, and genitourinary system. In addition, there are tertiary lymphoid organs. We should not say this as the organs, but they are the aggregation of the lymphocytes. They act as the tertiary lymphoid tissues. Okay, the most important tertiary lymphoid tissues are cutaneous associated lymphoid tissues. First of all, we are going to see the primary lymphoid organs. We have already seen that thymus and bone marrow are the primary lymphoid organs. Thymus, we are going to see the thymus first. Thymus is the very specialized primary lymphoid organ of the immune system. Within the thymus, T cells mature. It resembles a thymic leaf, so that it is called as thymus. So, it occurs its name thymus as it resembles the leaf called thyme leaf. எப்படி நம்மளுக்கு வந்து வாழை இலை இருக்குதோ அதே போல் தைம் லீஃப் அதே மாதிரி இருக்கும். தைம் லீஃப் ஷேப்பில் இருந்ததுனால இதுக்கு வந்து தைமஸ் அப்படின்னு they named it as the thymus. It is situated just below the thyroid gland. So, you can see in this diagram, it is situated just below the thyroid gland and above the heart in the thorax region as the derivative of the pharynx. So, where it occurs just below the thyroid gland and above the heart in the thorax region as the derivative of the pharynx. It begins its development in the 6th week of gestation period. In the fetal stage itself, it starts its development in the 6th week of the gestation period. It's quite large in size at the time of the birth, but it keeps reducing with age. So it starts degenerating after the birth. The thymus is the bilobed structure. So it is the bilobed structure. We have already seen it. It resembles the thyme leaf. It is the bilobed structure, and the two lobes are covered by the fibrous capsule. And each lobe is organized into small lobules which are separated from each other by the interlobular septum, that is called trabeculae. Each lobule is organized into two compartments, that is the outer cortex and the inner medulla. Two compartments, and the intermediate regions, that the cortico-medullary regions, also present. First we will see about the outer portions, that is the cortex portions. The cortex portion usually consists of immature T lymphocytes. They are otherwise called as thymocytes. And then reticular epithelial cells and few macrophages. They are all present in the cortex region. So in the cortex region, immature T cells, otherwise called as, they are thymocytes, they are present, and reticular epithelial cells are present, and a few macrophages are also present. As the T cells mature, they move from the cortex into the medulla. So the maturation starts in the cortex portion of the thymus. The reticular cells, the cortex also contain the reticular epithelial cells. So these reticular cells, they form the three-dimensional network structure, and in this meshes the thymocytes are found. The reticular epithelial cells are also called as nurse cells, as they nurse the thymocytes, that means the immature T cells during its maturation. In thymus, thymocytes, they undergo very rapid proliferations. The rapid cell division is at the same time, the rapid cell division is coupled with high rate of cell death, so that finally very small amount of the matured T cells migrate from the cortex to the medullary regions after thymic educations. In next slide we will see what is thymic education. So there are so much proliferations, production of the thymocytes and maturation of the thymocytes occur in the T cells, but only very small amount of the T cells migrate to the medullary regions after the thymic education. On the way to medulla, thymocytes begins to express receptors for antigens and other surface markers. Okay, so the immunocompetent cells only have the receptors for antigens and surface markers. Next, this T cells, the thymocytes after maturation, they are crossing the corticomedullary junctions and enter the medullary regions. So the what is corticomedullary regions and what is its functions? The corticomedullary region is the site where the selection of right T cells occurs. The selection is mainly carried out by the education. T cells undergo thymic educations through positive and negative selections. What is positive and what is negative selections? We will see. T cells either self-reactive, that means those cells which cannot discriminate self and non-self, they are eliminated from maturation, and the T cells which do not recognize foreign antigens displayed on the self major histocompatibility complex, that means MHC, they are also deleted. So this type of deletion is called the negative selection. What is positive selection? Only the T cells whose receptors, we have already told that the receptors they are having for antigens and other cells. The receptors recognizes the foreign antigens plus self-MHC molecule are allowed to mature or enter the medulla. This is called the positive selections. So this process is called as the thymic education. So this is the cross-sectional part of the thymus. So thymus is, it is the capsules, that is the thymus is covered by the capsules and it is separated by the lobes, and these are by the septae. They are separated by the septae. In the medullary regions they are having thymocytes. Thymocytes are immature T cells, and the cortical epithelial cells, reticular epithelial cells, otherwise called as the nurse cells, and some macrophages are present in the medullary cortical regions. And then medullary regions, the interdigitating dendritic cells, they are having the MHC2 on its surface, and then Hassel corpuscles, the wall of tissues, the Hassel corpuscles, their function is not yet known clearly, and the very few mature T cells are found in the medullary regions. So in the cortex regions, so many immature thymocytes are present, but in the medullary regions only small amount of the mature T cells are found. Then the medullary region, we will see about the medullary regions. The medulla consists of the vascular structure, more reticular epithelial cells, and scattered very few mature T cells. These are also some interdigitating cells. They are associated with the epithelial network, and these cells are rich in the MHC class II antigens. Then Hassel corpuscles I have already shown in this figure. So the Hassel corpuscles, I have already told you that Hassel corpuscles, they are the whorls of epithelial cells. They are around the central core of degenerating epithelial cells with unknown functions. They are also present in the medullary regions. So the cortical regions and corticomedullary junctions, after that medullary regions. So three regions they are thymus is having. And then the T lymphocytes are mature but non-functional in the thymus, because they are inside the thymus. The antigens cannot enter because of the capsules around it. The antigens cannot enter. So the antigens, the antibodies are matured, immunocompetent, but they cannot interact with the antigens in the primary lymphoid organs. These non-functional, they are non-functional T lymphocytes. These non-functional T lymphocytes travel through the blood or lymph circulations and finally reach the thymus dependent areas of the secondary lymphoid organs, and they get colonized there. Till they meet the antigens, they get colonized there. In the secondary lymphoid organs or T cells become, in the secondary lymphoid organs, these T cells, they become functional by the antigenic stimulations. So till they meet the antigens, they are non-functional, but after they meet the antigens in the secondary lymphoid organs, they start its functions. That means they react with the antigens and they kill the antigens. Then what are the main functions of the thymus? Thymus is the main site for T cell maturation and proliferation. The second one, it brings about the cell mediated immunity, and it is responsible for the graft rejection. So far we have seen about the thymus. Then thymectomy. What is called thymectomy? Thymectomy means removal of the thymus. So in the experiments, some of the scientists, few scientists, they removed thymus from the rat and found the consequences of the thymectomy. So removal of the thymus in mice results in the impairment of the cell mediated immunity. So it results in the drastic change in allograft rejections, delayed hypersensitivity reaction, and it also results, the thymectomy also results in the wasting syndrome. That means wasting syndrome leads to the hunched appearance, ruffled fur and diarrhea. and immunodeficiency also results in the thymectomy. So in humans, congenital birth defect, that means DiGeorge syndrome. The symptoms of the DiGeorge syndrome is delaying in the learning to walk or talk and other developmental and learning process that results because of the poor development of the thymus. The next primary lymphoid organ is the bone marrow. The bone marrow is the major hematopoietic cellular connective tissue within the bone and produces all immune cells and delivers them in the circulations. That means hematopoiesis means synthesis, hem means blood. So blood synthesis, all the blood cells are synthesized by the bone marrow and they are they delivered to the circulations, to their sites through the circulation. There are three types of the bone marrow: red marrow, white marrow and stroma. The red marrow is richly supplied with blood. Usually the bone marrow is richly supplied with blood vessels and capillaries, but it lacks lymphatic vessels. You must know that it do not have any lymphatic vessels, but they are supplied with blood vessels and capillaries. First of all, we are going to see about the red bone marrow. Red marrow, it is called red marrow because its color is red because of the high blood flow or presence of the red blood cells and its precursors. It is the main site of the hematopoiesis, that means blood cell synthesis. At birth, all bone marrow are red in color. With increasing age, almost half of it is converted into yellow marrow because of the deposition of the fat. So yellow marrow because of the presence of the fat cells, they found in the interior of the long bones. They reduced hematopoiesis. They are here reduced hematopoiesis only occur. These two forms, the red marrow and yellow marrow, are interconvertible as demands change. If we need more blood, then yellow marrow can be converted into red marrow to synthesize more blood cells. So if there is no demand, the fat cells accumulate and they form the part of the red marrow will become the yellow marrow. Then stroma, it is the tissue of the bone marrow. It provides the microenvironment of the hematopoiesis. Cytokines and growth factors are produced in the stroma only. They supply iron to the RBC. Okay, so stroma, it is the tissue of bone marrow. It provides the microenvironment for the blood cell synthesis, that is otherwise called hematopoiesis, and the cytokines and growth factors are also synthesized in the stroma. This stroma supply iron to the RBC. What are the functions of the bone marrow? The functions, you see, it's the main site of the blood cell formations. All the blood cells are formed in the bone marrow. Their functions both as primary as well as secondary lymphoid organs, that we will see how later. Then B cells originate and mature in the bone marrow. B lymphocytes, they originate and mature in the bone marrow. Then selection process occurs that deletes those B cells which are self-reactive. So in the bone marrow itself, the selection process occurs. The B cells which are recognizing self as the non-self are deleted from the circulation. Mature B cells exit via vascular sinuses and enter the bloodstream. Some of the lymphoid stem cells, they migrate to the thymus and they mature into T cells. Actually, all lymphocytes are produced by the stem cells only, and a part of the lymphocytes are migrate to the thymus and there they mature into T lymphocytes, and the remaining part of the lymphocytes in the bone marrow, they mature into B lymphocytes. Cytokines and growth factors are produced in the stroma of the bone marrow. Bone marrow is also the source of the antibody synthesis. Then bone marrow functions as both the primary and secondary lymphoid organs. How? Bone marrow functions as both the primary and secondary lymphoid organs as B cells are developed and differentiated into immunocompetent B cells. So it acts as the primary lymphoid organs as the B lymphocytes mature and proliferate in the bone marrow. So it acts as the primary lymphoid organs by doing this work. And then in mammals, few days after the secondary response, primary response and secondary, after the secondary response, the activated memory B cells, they migrate to the bone marrow where they mature into plasma cells, and in these plasma cells they produce antibodies against that particular antigens. So bone marrow, they can act as both primary lymphoid organs as well as the secondary lymphoid organs. Thank you.